Antiparallel beta structures comprise the second major class of protein conformations. In these, the antiparallel beta strands are usually arranged in two beta sheet layers that pack against each other and form a distorted beta structure, the core of molecule. Depending on the way the beta strands around betas are connected along the polypeptide chain, they are divided into three main groups: up and down betas, Greek key beta, and jelly roll beta. And recent advancement just lead to second class in this category, that will be circular beta or beta helix. We are looking at superoxide dismutase, where 8 anti-parallel betas are making the barrel shape. This is just a canonical some model. You can see the neighboring two betas just anti-parallel. Okay, two layers, four betas in the front and the other four at the opposite side. This is top view. This is real protein. Betas are making certain curvature. Basically, in this model, antiparallel betas ranging 4 to 10, or sometimes more than 10. They just often make barrel shape, and then the basic motif in this structure will be beta loop beta. So in this slide, you are comparing up and down barrel at the left side with thin barrel at the right side. So when you look at the topology, betas in thin barrel, they are parallel, whereas betas in up and down barrel, they are anti-parallel. So just connected by a loop in up and down barrel. However, in thin barrel, they just connected by a loop, helix and loop. But they both shares a barrel shape of the structure. This is another protein example called human plasma retinol binding protein, RBP. It carries vitamin A, retinol, from the liver, where it is synthesized to the other cells where it is consumed. So you see 8 beta antiparallels making barrels. Just inside there is some open pocket where vitamin A, which actually lipophilic types, so surrounding the protein portion also must be hydrophobic. This shows you retinol A in yellow color, and surrounding residues of RBP. They are pretty much hydrophobic residues. How this kind of structures could be made? Actually, when they are uh, this protein RBP is fold into this kind of structure, requiring the retinol from the beginning. So RBP, when aligned for their beta ability sheet, uh, every the other alternating residues, they just shows highly conserved hydrophobic residues, meaning each has a facing, every the other residues just face the same direction, heading to retinol A, making hydrophobic interactions with uh, ligand. There has been known two kinds in this class. First, RBP superfamily. It has 8 beta sheet. You can see the proteins belonging to this class. The second group will be P2 family, which comprise 10 beta sheets. And again, you can see the proteins in this category. Both are a lot different for their primary structure. In other words, they don't share any primary structures, but the 3Ds are very similar. We're gonna explain the coat protein of influenza virus. There are two types of the coat protein, yellow and the green one. Before we learn about this protein, I'm going to just explain briefly the life cycle of this virus, because the function of coat proteins must be explained in terms of life cycle. Virus actually adsorb to the surface protein of host cell, and then it could enter into host cell by endocytosis. Inside the cell, as a result of endocytosis, it is surrounded by a membrane called endosome. So for this virus to multiply, it must escape from the surrounding membranes. That process is called uncoating. And then right now this virus is ready to multiply. Okay, so as usual, DNA could be copied, replicated, and also together some the viral proteins could be made, and they assemble each other into daughter patch, and then daughter virus, it could escape. In this case, as mode of budding on membrane. So released virus is encapsulated by membrane. Two membrane protein on influenza virus are neuraminidase, and hemagglutinin. We're going to look at the neuraminidase first. The extra the membrane portion head portion was cleaved by treatment of pronates, and just structure was determined, showing this kind of four anti-betas. Topology clearly shows four anti-parallel betas. Actually, head is composed of 24 of this kind of four anti-parallel beta. So you are looking at first portion of the whole structure. So how many? 6 four anti-parallel betas are arranged in a way. First one meet with 6, something like a circular barrel shape. Okay, so it is called like a 6-bladed propeller structure. In topology at the bottom shows the relationship. They just each unit just arranged by side by, but the last one is meeting with first one. Okay, the whole picture for the head of neuraminidase are shown in this slide. Okay, so actually they forms a four-fold tetramer. Okay, so this is each unit again by side view. This is just a simplified version for this structure. Okay, one side of end showing the long stretch of loops where active sites actually positioned. Compared with the other side, just short loops are connecting the betas. This active site actually composed of 12 loops, and it could cleave cyclic exit of both viral HA, which I'm going to explain soon, and the host membrane proteins. What is the role of cleavage of cyclic exit? It could help the progeny of virus from escaping from the host cells where it was produced.